Yep, we're going to be talking about hacking structural biology with Python and Pandas. I know you guys aren't biology experts, probably, and that's fine. But hopefully I want to inspire in you, maybe an interest and let you realize that maybe this is all within your fingertips. You could be a structural biology hacker hobbyist in your spare time. So in this notebook, we're just going to scroll through and take you through a real research story, going from generating the data and analyzing it to coming up with some, new discoveries about something that's inside of our bodies. So mind the imports. I'm also going to execute, as we go through. This is live. It's a true live demonstration. So mind the imports. This talk wouldn't be anywhere without these great imports, but I'll mention those at the end. Some colors also, nothing to worry about. Let's first ask the general question, what are proteins and what is structural biology? You guys might know about the genome. Everybody's getting genetic sequencing these days. But you might not know that a lot of the genome encodes for proteins. So your body has some mechanism that reads off this sequence of G's, C's, T's, and A's and converts it into this long string of amino acids. Amino acids make up this thing called the protein. And it folds into this funky shape, which is gives it this structure. So that's what I mean when I say structural biology. It's these little machines in our body that exist and do some task. So this one I'm showing, I should say that, it's just made of atoms. This little representation showed their little balls like hydrogens, carbons, oxygens, nitrogens. And that really gives it this structure. So we show it sometimes as this ribbon, this like, this long stringy thing with colors, but we can show it in many ways. But as you see, this view here gives you a little more information. It's less cluttered. So this thing I'm showing you is a protein called lysozyme, and you've probably eaten it when you consume an egg. About 4% of the protein in an egg white is this protein. It's a natural antibiotic. It kills bacteria. So that's its sole job. So it does that by this little cleft here. This little cleft inside the protein. Anytime it makes in contact with the cell wall of a bacteria, it starts slicing. It starts breaking the chemical bonds of these sugars that are on the cell wall. And it ends up breaking the cells of bacteria, killing them. So it's the goal of me as a scientist to understand how this structure relates to this function and how diseases and drugs might affect this relationship as well. But looking at the structure of proteins is a cool thing we can do, but we can also study how they move with supercomputers. We can study how these proteins fluctuate with real time, making computer models. So I'm a computer modeler. We do this thing called molecular dynamics. How do you do that? It's a 3-step process to do to take this and do a simulation to watch it move. So thankfully, a lot of effort for decades and decades has been compiled into one database online called the Protein Data Bank, which is where I pulled this protein from. And if we didn't know what the sequence or what the structure was, we could make it ourselves. If you've ever taken a chemistry class, you might build a molecular model. It's very similar on computer. Anyway, this time we're going to, after you have your model, you define the interactions that atoms might feel. So that would be deciding, how strong is a bond between two atoms. And so that requires a little bit of domain expertise. You may have to read a few papers in order to know about this step. But there are also some really, really good defaults. Like once you read a bunch of papers, you start to realize the defaults. Like everyone has a similar methods section of their paper when they're studying, a certain class of proteins. And then finally, as your eye probably drew to right away, we can do simulations like this dog is doing. The analogy I'm trying to go for here is that the dog is imparting these velocities on these atoms, these balls, and moving them around with real physics. So it's not actually so different in a computer simulation. We're doing pretty much what the dog does when we execute a supercomputer simulation. We initiate velocities and start watching these particles bump into each other. And that's what I do on a massive scale. But let's use the power of Python to go through these steps, building a model, defining some interactions, and running a little simulation right inside this Jupyter notebook. So some great work done out of Stanford has built these tools which allows us to do that information. So I'm gonna run this cell here, which is gonna pull a protein. This is the same protein I showed you here from the protein data bank. And I'm going to fix it up a little bit. It was a little bit broken. It was missing some atoms. And I'm just going to just do that, execute that. And the next step would be loading it into this. So I should say I'm not going to expect you to know every single line that's going on here. This is really more just a general scope of this thing. So what we're going to do is in this whole block of code here, we're going to define those interactions. That was this step here. Define the parameters and interactions. That's called the force field. We're going to define some other things like what temperature are we going to do the simulation at. The next step would be to define how we're going to do the computation. I'm going to use OpenCL, but you could use CUDA if you had some GPU, or you could use CPU, of course. And we're going to set some velocities, just like the dog did, and perform the simulation. I should have been doing it already, because it takes a second. It takes more than a second. It takes an estimate. It's going to take 27 seconds. So in the browser, I'm interacting with, this is actually a Python API to some C code. It's not written in Python, because it's high performance code. And this is running the same thing that I would do on a supercomputer right in the browser on my MacBook here. And so what do these numbers mean? Well, this is the progress of the simulation, and this is how many steps we've performed. Steps of simulation would be an incremental step in real time. But proteins work on a very fast time scale. Like femtoseconds of time is what we're working with here. Every time I step one step ahead, I go move atoms a tiny little bit more. So, and that all adds up. We can do a long simulation over months and really gain a lot of data to understand how these proteins move. But let's just look at what we built there. So this is a cool IPython notebook widget that shows this lysozyme protein. There's that cleft. And I can even animate it with this data that we simulated. And you can even zoom in. So not a lot is going on. It's little tiny fluctuations here and there. You may have to run a longer simulation in order to get some juicy results out of this. You may even have to add a drug or mutate it or change the protein in order to learn some new things in science. But that's the basic concept. Just done it right before your eyes. So take a step back and think about what our data is in this simulation. It's not really a fancy picture and 3D model. It's really just an array. If we look at this data I generated, it's called a trajectory, and look at the shape of this, and look at this data, it's really just a three-dimensional array. Where on one axis, it's the number of time steps. So here I did 7,500, but I really saved data every 100 time steps. And I missed the first one for some reason. So I have 74 frames of data in time, and I have 1,958 atoms. If you can't really tell how many atoms are in it, you can view it in this form, which shows you that 1,900 atoms. So that's their protein. And then the three axis is just the x, y, and z coordinates. So again, that's the number of time steps on one axis, the number of atoms on the other, and the dimensions of space, x, y, and z. That's our raw data. We fill up trajectories that have sizes of terabytes with just these coordinates of atoms. And so you can imagine there's a lot of processing involved in this because you don't often care about every single atom in our box. But let's now jump out a little bit, jump to a bigger scale. Because this is a small protein that's cool and interesting, but I study for my PhD thesis something a bit more, I don't know, I shouldn't say exciting, shouldn't want to say some proteins are less exciting than others, but this one's pretty cool. So that's why I'm working on it for several years. So in your brain you have neurons, and you may know that neurons fire, and neurons initiate these electrical impulses in our body. Any motion, any heart contraction, any muscle contraction. They're not necessarily neurons, but they are electrical impulses in our body. And those are a result of ions. What are ions? Ions are atoms that have little charge, like sodium, potassium, and calcium are charged atoms on the periodic table that float around our body, and they're used inside cells. So inside the cell wall, represented here, are little holes, channels. Really, they're just little cylinders that allow ions to flow through. And they do that in a selective way. They say, okay, you can come in, but you can't come in. You can come in, oh, you're fine, you're cool, you're not cool. So they initiate this net charge, because these little things have charge, so that there's charge on one side and no charge on the other, that's a voltage. And this voltage propagates through our body. It's really amazing. But I look at the molecular detail, the tiny detail of each protein. So this is the protein that lets ions inside the cell. This is the ion channel, as it's called. And so it's a little more complicated to build a model of an ion channel because you have to put that membrane in. That's the yellow stuff here. So you build a little patch, a little box that has a little bit of membrane and then the protein in the middle, and then you scatter some ions around the box just randomly. It gives you an algorithm. And then you fill it with water. So this shows you the step-by-step building of this molecular dynamics simulation. And so in this system, there's not 1,900 atoms. There's something like 160,000. So a few months of supercomputer simulation later, simulating this puppy, and we can have a nice juicy data set. Let's just, you can even visualize it. So this is the protein here, and this is a top-down view. So we could zoom in here and pretend we are an ion. And we're descending inside the channel. I'll zoom in. Descending inside. And these little red balls are the sodium ions themselves. So don't ignore those long, that's a rendering artifact, those long things sticking out there. That's not real protein. But this is the thing. Now the water is not shown and the lipid bilayer is not shown. But that's the gist of it. It is a protein where ions flow inside and out. And I can even render that for you. I can even animate that for you. And we can take a side view and really see what's going on. So there is a hole in the middle of it, but it seems, and the ions are free to move, but they tend to stick around, bunch up in this one zone. And why do the ions stay in one zone? Well, it's energy favorable for them. If you're a charged atom, like a sodium atom that has a charge of +1, you want to be with something of the opposite charge, because opposite charges attract. So if you're positively charged, you want to be with something negatively charged. So that's what that part of the protein must be negative. But we'll dig into that a little bit later. So far, we haven't really done any NumPy or any pandas, or use a lot of the functionality that's built into Python other than executing the code, executing the simulation. So let's do that. Let's just get in the meat of it, because we need to write a scientific paper about this. So what's our first step? Well, I'm going to reduce the complexity of the system a little bit. If I zoom out of this system, you can see that there are ions scattered all over the place. And although it's probably not too many of them, I'm only care about a certain number. In this case, I only really care about 2, because 2 are the only ones that are inside. Well, maybe 3, that one is. So I can do a little geometry in order to take this gumball view, shows the position of all sodium ions over all time in my simulation. And you can see that it samples pretty much all the positions, except for where the cell wall would be. And I'm going to just throw away a lot of that data. Just simplify my life a little bit. So I'm taking that 3-dimensional array, or sliced Rubik's Cube, if you like my graphics, and I'm going to just slice out some of on the number of atoms axis that I don't care about. So I'm going to make this cylinder and just do some really basic algebra to find stuff inside a cylinder. So this is some specific code for analyzing simulation trajectories that just grabs the positions of selected atoms. So I'm going to define what's called the center of mass, or center of the cylinder. And it actually changes at every time step. So I actually have this reference point that's going to define where the center of my cylinder is. And it has 3 coordinates, x, y, and z. And 501 was the number of time steps that I have in this simulation movie. So I have a 501 by 3 array. And then I'm going to grab the positions of all sodium atoms. That's Na, if you know your periodic table. So I'm going to give me all the name NAs in the box. And I'm going to subtract off the reference. And NumPy is nice because you can do some little broadcasting here in order to get that. Next, do this Boolean check. Give me an array that shows me at what time was a particular atom inside the cylinder. In this little summary shot, you can see it was only true once. So that was not the 0th atom, but the first atom, which is this row. It was inside the cylinder at time 498, 499. And so that was right there was a true. So you can use NumPy's where, kind of a cool thing, to get the indices of these trues. That's what this np.where is. So you can get those indices and it looks something like this. So the first atom was inside the cylinder at this time. And then we can use that to construct the reduced data set, the reduced table of all the sodium atoms we care about. So that's this table here, where each row represents an atom at a specific time, and it has a specific ID. Call that the res ID for some reason. And so that's the identifier for the particular sodium atom, because remember, there's lots of sodium atoms in the box. There's like 47 of them. And yeah, we have this data set. But, I'm going to cheat a little bit. And to make things juicier, I'm going to not just analyze this one simulation that I loaded in. So sorry, earlier I loaded this simulation. I'm going to load up now 20 of them and have a higher resolution of data. So this is like now a multi-gigabyte data set. I'm just going to load it into a data frame and show you what that looks like. So here's the same data set, just a lot more rows. 7 columns, but a bunch more rows. Okay, so now we have this nice data set of the positions of all ions in the channel. Let's take a look at that. So if we zoom in on this particular region where a lot of atoms were sticking around, we see that, it's interesting. There's actually a bit of protein sticking out into this zone. We'll revisit that later in the talk. But I mainly care about just the ions right now, so I'm going to label them with color. Say that, if you're the first one in the channel, you're the red man, you're the red ion. If you're the second one, you're the green. If you're third, you're blue. Red, green, and blue ordering. Now, it's important to order them to, help our feeble minds understand what's going on here, because they swap places a lot too. In this particular channel, they swap places, and you start getting a color mess. So the goal that I want to look at first is just to plot the positions of these ions along this axis called Z versus time. Time series of ordered ions in the channel. And so finally you can use, this is pretty sweet. So I'm using the same data set we looked at before to plot this information. You could think I could just plot the Z column of this data frame. Which is fine, but we might not get the ordering. We want to color these data points. If this was a scatter plot, that's cool. I could plot a scatter plot of Z, which would show you over time where the positions of all these atoms are. But it'd be great if we could color them. So I want this column that I just miraculously did by executing this cell that didn't explain, which is called order, which would say, okay, you're the deepest in the channel, now you're the red ion, you're the order 1, or order 0 or order 1, or if there was 3 ions, actually, I want to rank them by Z. I want to sort within each time step. So at time step 1000 in this example, there were 4 atoms in the channel, and I want to rank them, 0, 1, 2, 3. And the algorithm to do that is like in a nice, you could imagine that you could break up this table. Like, what would be the algorithm that you might suggest to get that column? Okay, you could do if you know a bit of pandas, you could group by the frame value. You could say, give me all the rows that have the frame value of 1, and then I could sort of re-index it. I could say, okay, we'll sort it, then re-index it, and reassemble it. But the performance of that is pretty painful, and then you have to join them all together. I actually don't know a good way to do it using pure pandas, but I do know a cool way to do it in NumPy based on Stack Overflow wizardry. I couldn't come up with this myself, but I might not even explain it. You can but okay, I will explain it because it's pretty cool. So for some reason, you make this column called total count, which is an incrementing number from 0 to the number of rows. But you also have this column called the repeated cumulative sum. So if I was at this time step, here over all the times, I have the total number of ions in the channel. Anyway, I subtract this column by this column, and you get the ordering column. Let's not drill too much into the detail. Let's just move on to the fun stuff. So I've delayed the payoff long enough. This is what it looks like to look at how ions enter this channel in and out. The red one is the first. It enters the channel. Then all of a sudden green comes along, bounces you inside, and you start hanging out of this zone. And then when you least expect it, the blue ion approaches. And does it really bump the green one in? No, a little bit. Knocks him down over here. And then he just leaves all of a sudden. So computer simulations, you don't know what's going to happen. Things happen randomly. In fact, I could plot this. I did this experiment 20 times, the same simulation. And every time it's a little bit different. So we want to look at averages. I want to say, on average, where are you the most time? I spend a lot of my time maybe here in this zone. And it's because you can see lots of red clumping up there. And a lot of time over here. And so we want to annotate what parts of the channel are favorable for an ion to be at. We need to sort of label who an ion's talking to. By default, an ion talks to about 6 different people. Not people, other atoms. It talks to 6 different waters when it's floating around in the water. But sometimes when it's in the protein, it exchanges one of those waters for protein. It says, okay, I don't want to talk to water anymore. This protein looks nice. I want to talk to protein. So I'm going to say, if this ion is talking to these orange bits of protein, then I'm going to label this ion orange. And if this ion is talking to yellow, I'm going to label it yellow. And if this ion is talking to pink, I'll label it pink. So here we can do, I'm going to load up and cheat a little bit and load up another table of data that is the same, but it has extra columns that says who are you talking to. For every ion, what are you talking to in the data set? So this thing called orange is this column, E177. That's just the protein code for it. So as you see later in the simulation, this column here, you were talking to 2 of those. And then this column here is number of waters. So I'm talking to 2 protein and 3 waters. And so I can make a code about who you're talking to. I can make, and then group by that code and plot each histogram separately. So it's easier if I just show you the picture first. So this is the histogram of the positions of all atoms in the channel. So this side is outside the channel, and this side is inside the channel. And so as you see, we pass by orange first a little bit, then we talk to yellow, and then we talk to pink the most. So that kind of agrees with the picture we saw here about who you're talking to. Okay. And this is our baseline. Remember, this is our we want to study a lot of things about this channel, like maybe how diseases affect it, and how drugs affect it, and all these things. But we need a baseline. And so once we have our baseline, then we can really start to have fun. That's when your PhD magnifies in complexity. So I do another experiment where I notice, this protein is actually pretty flexible. Sometimes that orange guy, it's colored differently in this picture, is sometimes, facing up, and sometimes it's facing down. So notice there's some flexibility there. So, what's going on? Is that why maybe we have 2 peaks here? So you can quantify this up and down based on an angle. We'll get into the details of the angle stuff, but you can plot this angle, ups and downs, upsies and downsies. I load up this table that has angle information, and you see that half the time the protein's in up, half the time the protein's down. So there's a split. So what if we did another computer simulation where we were like, nope, you're not allowed to flip up. You're not allowed to flip down. You can only stay one way. It's a trick in computers you can do because you can change everything specifically to your liking. You can run a computer experiment that would not happen in nature necessarily, but you could just learn. You could use this to learn. We're in academia after all. We can just explore. So we did an experiment again, the same one where we restricted if the protein can move. We said, can this protein flip? We'll make it so the protein can't flip between states. And here's the same time series showing this angle, this up and down angle. No, it's not able to go in the other state. So what does that look like? So what does this distribution look like when you do that? And that's kind of a dramatic difference. Oops, executed that cell already. That's this. So what we did is we restricted protein flexibility, totally disrupted, where ions stay inside the channel. What's going on? Keep an eye out for future papers, but it's pretty cool that, we learned that this is so critical. This protein flexibility at this point is critical. If you were born with a mutation that had no atom there, a different bit of protein there, your ion channels might not work. And that's all. That's all I have to say. A little sneak peek into my research. I'd like to thank the supercomputing team, the funding sources, University of Toronto SickKids, Python Canada, and all the Python libraries that made this possible. Because without Python, you don't even want to know how long this would have taken. Okay, that's it. Okay, so if you have a question, you can have to come up here, or I can repeat your question. But I would be happy to take any question, no matter how basic, how advanced, about how Python comes into my work, or how biology can be studied with Python. Yes?
Okay, so I have two questions. The first is, could you give a little bit of an overview of the Python libraries that were used in this?
Sure.
You sort of mentioned that at the start.
Yep, the question was what Python libraries were used for this. So most of the normal scientific stack of software was used, like NumPy and Pandas, and Matplotlib for the plotting. But the only ones specific to molecular dynamics, which were in the first few cells, were these ones called mdtraj and OpenMM. And those are on the slide deck as on the website omnia.md, and that's mainly from that group at Stanford that made those. So after a bit, like after you generate the data, there's no more biology anymore. It's just data. Data's data. We're all the same after we get just a bunch of pandas tables, data frames. So those are the only ones specific, and those are for generating the data. But once you, the analysis of the data is the traditional tools.
And then, sorry, the other question that I had really quickly was, how useful would this sort of approach be for predicting binding sites of transcription factors or other nucleotide binding approaches?
Yeah, the question was, how useful would this approach be for predicting binding sites? Like proteins attach to each other a lot. Proteins and RNAs, all the things that live inside your cells. There's lots of binding. This underlies a lot of how our bodies work. And this is, a great approach for that. But some things happen on the biological time. Some things take, a second. Some things take a long time. A second is eons. It's outside of the reach of supercomputers for most of the part. A lot of the things we study happen on a shorter time scale. And so, like, an ion going through a channel is pretty fast compared to maybe some of this other complicated biology stuff. So there are some problems where it works and some where it doesn't. But it's overall a great approach for that.
A follow-on for that, if you scroll down, there was another package, SimKit or something?
Yep, SimTK, oh yeah, you're right. I imported them here where I use them. Yep, SimTK is part of that is the OpenMM package. That's what performs the simulation. That's what does the grunt work on OpenCL, CUDA, or CPU. Oh, yes.
So how low does the simulation run in terms of modeling the physics of chemistry? Is it a lot of heuristics, or do you use automata or anything like that?
The question was, how low level, like science or physics are we doing? Yeah, we only go as low as, I showed the dog because it's pretty simple. The physics is really Newton's equations, but a little bit more, a tiny bit of stuff layered on top. I mean, you could do a computer simulation with quantum effects, built in. Well, in fact, that was the Nobel Prize in chemistry, I think 1 year ago, was for a computer simulation, for computer discovery or the use of simulations that incorporate quantum effects. So I don't do those simulations because they take extra long. But these ones, you make some approximations. You kind of glaze over a little bit of details. Atoms are just point masses, pretty much, that have some kind of radius of interaction and a charge. So that's kind of like the level of coarseness. You can go full scale. In academia, everyone goes, you can zoom in and out.
And the other question I had was, do you use any of these notebooks in your papers or sharing information with other scientists?
Yeah, that's in the progress. It's not really standard in my field to do to distribute a notebook like this with a paper, but it's definitely moving in that direction. And I'm going to lead the force to do that. I'm going to lead the troops. Say this is how it should be done. So.
I'm afraid we've run out of time. Chris, would you like to entertain any other questions in the hallway afterwards?
Of course. Yeah. Just
Okay,
come up to me anytime today.
So if you have any further questions, Chris will be around, potentially in the hallway outside. The next session in here is in about 5 minutes. And you know what? I've now managed to forget it as I walked up here. All right. Thank you, Chris, so much.
You're welcome.
It was very informative, everyone.